Good morning, all of you. This is Sujata Sharma, Assistant Professor of Zoology from Government College Chichroli. Again here to explain you next topic, that is digestion. Now digestion is related to very important activity. We call it nutrition. So what is nutrition? Nutrition refers to sum total of all the activities by which organisms obtain and utilize the food particles. If I say in living world, there are three types of nutrition: autotrophic, heterotrophic and mixotropic. In autotrophic nutrition, organisms prepare their own food by using a source of energy, and source of energy can be light energy or chemical energy. So depending upon that, autotrophic nutrition can be photosynthesis or chemosynthesis. As the name indicate, in photosynthesis, organisms use light energy for preparing their food. They use light energy for converting inorganic raw materials like carbon dioxide and water into organic form, that is in carbohydrates. All green plants, algae and photosynthetic bacteria, they are capable of showing photosynthesis as they capture light energy due to chlorophyll pigments they carry. Regarding chemosynthesis, if I say, it is use of chemical energy for preparing their own food. There are number of bacteria in nature, particularly nitrifying bacteria, iron bacteria and sulfur bacteria. They help in mineral cycling, and during that activity they oxidize number of chemical compounds, and these oxidation reactions are releasing chemical energy. That chemical energy is utilized by bacteria for manufacturing their food. Like nitrifying bacteria, these are very important bacteria for nitrogen cycle. They help in nitrification activity. Nitrosomonas, Nitrosococcus and Nitrocystis. What they do? They convert ammonia to nitrites and then to nitrates during nitrogen cycle. And this conversion of ammonia to nitrites and then oxidation of nitrites to nitrates release chemical energy. That energy is utilized by bacteria for manufacturing their food, for producing organic compounds. And since they are dependent upon chemical energy, this mode of nutrition, that is autotrophic nutrition, we are calling it chemosynthesis. Now second type of nutrition is heterotrophic nutrition, where organisms are dependent on others, plants or animals, for obtaining their food. They can be Holozoic or holotrophic organisms. They can be parasitic or they can be even Ceprotrophic. Now in Holozoic or holotrophic nutrition, organisms, they ingest and break down solid food inside their body. And since they are breaking the solid food, so they are releasing enzymes inside their bodies. With the help of enzymes, they either consume whole organisms or their parts. If I say examples, all free living animals, whether it is amoeba, which is unicellular, hydra or even man, these all are holozoic or holotrophic animals. Now depending upon type of food that holozoic animals are consuming, they are of several types. So types of holozoics or holotrophs are, it is herbivores when they consume plant matter. Cows, buffaloes, goats, zebras, these are herbivores. If they consume flesh or animal matter, it is said to be carnivores. Tiger, lion, leopard, these all are carnivores. If they consume both, like man, crow and bear, they are omnivore. They consume both plant as well as animal matter. If holozoics consume fruits, animals which consumes fruit, we call them frugivores, fruit eaters. Birds and monkeys are very common examples. If holozoics are dependent upon dead and decaying matter, if they consume detritus, dead or decaying matter present in the soil, we are calling them detritivores. Example, earthworm. Insectivores, these are the other types. As the name indicate, they trap insects for fulfilling their requirements. Insectivorous plants, these are Nepenthes like pitcher plant, Venus flytrap or even Utricularia. These are insectivores. They consume insects, they trap insects just to fulfill their nitrogen deficiency. Then in nature there exist granivores. Granivores are those animals which feeds on grains. Example, pigeon and many birds, they are granivores. Then which consume fishes, we call them piscivores. Birds and other animals, they are said to be piscivores. And even animals are filter feeders like whales, Herdmania. They are said to be filter feeders because they filter the water current for obtaining their food. They filter water current for obtaining zooplanktons. That is why the mode of nutrition is said to be filter feeding. So these are type of holozoics depending upon food they are consuming. Now second type of heterotrophic nutrition, it is parasitic. Parasitic means organism which is called parasite dependent upon host for getting the nourishment, and it consume ready-made food from the host body. If I say example, parasite of sleeping sickness, trypanosoma, very common example. Taenia solium, commonly called tapeworm, it is endoparasite. Ascaris or roundworm is another endoparasite, and even plant parasites exist in nature. Cuscuta is a parasitic plant which depends on other plants for getting the nutrition. So these are parasitic organisms, and the mode of nutrition is called parasitic heterotrophic nutrition. Now third type of heterotrophic nutrition is Ceprotrophic nutrition. As the name indicates, Cepro means rotten. Here organisms depend on dead or decaying organic matter. They depend on dead and decaying organic matter. In fact, they release their enzymes outside the bodies. They release enzymes to break down the organic matter outside their bodies, and they absorb it in the simple form. Very common examples, these are fungi like bread molds, mushrooms and bacteria in nature. They all are Ceprotrophic. Now third type of nutrition I told you, it is Mixotropic. What is Mixotropic? In Mixotropic nutrition, organisms show both kind of nutrition, autotrophic as well as heterotrophic. Very common example is Euglena. We know Euglena is a connecting link between plants and animals, kingdom Plantae and kingdom Animalia. We called it as a plant animal. Why it is plant animal? Because it has chloroplast on one hand and flagella on the other hand. So due to chloroplast, it is able to capture sunlight and perform photosynthesis in the presence of sunlight. But when sunlight is not there, then Euglena become heterotrophic. It start consuming other organisms. So as Euglena is autotrophic in the presence of sunlight and heterotrophic in the absence of sunlight, so we are calling it mixotroph, and this mode of nutrition we are calling it mixotrophic nutrition. Now we are going to talk about physiology of Holozoic nutrition. When we talk about physiology of Holozoic nutrition, there are 5 steps basically: ingestion, digestion, absorption, assimilation and egestion. Ingestion is intake of food particles. Digestion is the breakdown of food particles. Absorption is the activity by which digested food enters our blood and lymph. Utilization of food by cells and tissues, we call it assimilation. And the removal of undigestible remains, it is egestion. So, digestion is the activity which involves breakdown of food. Large, complex and insoluble food particles, they are changed into smaller, simpler and soluble ones by the process of digestion. There are 2 types of digestion basically, intracellular and extracellular. As the name indicates, intracellular means which occurs inside the cells as in amoeba, and extracellular, which is also called intercellular digestion, it occurs outside the cells, that is in digestive cavity. Very common example, humans, hydra and all higher animals including all the vertebrates. Let me tell you, planaria and hydra, these are 2 examples that show both kind of digestion. They are very unique, as hydra and planaria both show intracellular as well as extracellular digestion. Now when we talk about intracellular digestion in amoeba, it occurs inside the cell and we also call it phagocytosis. You can see in the picture. Amoeba has finger-like projections called pseudopodia, and through pseudopodia it is going to engulf the food particles like bacteria and other smaller microbes. Now those food particles, bacteria, they enter amoeba through a vacuole called food vacuole or phagosome. Inside food vacuole or phagosome, lysosomes become active and there start digestion of food within phagosome. From here, digested food particles, they are absorbed into cytoplasm, and finally undigestible remains, they are thrown out again through the cell membrane. So amoeba being unicellular, it shows all the activities within the cell. Ingestion by pseudopodia, digestion by food vacuole, absorption by cytoplasm, assimilation is the utilization of food by the cell for growth, repair and well-being, and egestion is the removal of undigestible remains. When we talk about digestion in humans, as I told you, we have extracellular digestion which occurs in our digestive tract, in our digestive tube, in our alimentary canal, and this involves mechanical as well as chemical breakdown of food particles. Mechanical breakdown basically occur by the action of teeth, movements of stomach, we call them churning movements, and segmental movements of intestine, while chemical digestion occur with the help of digestive enzymes. Let me tell you, all digestive enzymes produced inside alimentary canal, they are said to be hydrolases. They are hydrolases as they carry out breakdown of substrate or food particles in the presence of water. So all digestive enzymes, since they require water for breakdown, we call them hydrolases. So when we say digestion in human beings, digestion begins in buccal cavity and it completes in small intestine. What all events occur in our buccal cavity? The moment we ingest the food, there occur taste of food by tongue, the very first activity. Tongue is highly muscular organ and it bear taste buds on its surface. And those taste buds, they have gustatoreceptors or sensory cells which are there for the taste of food. So taste of food is the very first activity. Then there start grinding of food. We have heterodont dentition, 4 types of teeth, incisors, canine, premolars and molars, and they all help us to grind or chew the food particles in the buccal cavity. Along with that, there start release of saliva by our salivary glands. So this is the important fluid that begins digestion in our buccal cavity. Saliva is released by our salivary glands and we have 3 pairs of salivary glands in our buccal cavity. There is a gland, parotid gland, the largest salivary gland near ears on the sides of face. Then sublingual gland present below tongue and submandibular, also called submaxillary, present at the angle of lower jaw. So we have 3 pairs of salivary glands, parotid, sublingual, submandibular or submaxillary, and these all glands, they are releasing saliva. Saliva is a fluid, digestive fluid with pH 6.8, that is somewhat neutral or you can say slightly acidic. Its composition includes water about 99%. It has mucus. Electrolytes are also there. Sodium ion, chloride ion, potassium ion, bicarbonate ions, thiocyanate ions. These are present in saliva. And saliva contain 2 important enzymes, lysozyme and salivary amylase. Salivary amylase is also called ptyalin. Out of these 2 enzymes, lysozyme is antibacterial. If any bacterium or foreign particle is going to enter our buccal cavity with food, that foreign particle is killed by the action of lysozyme. But this enzyme, salivary amylase, it is highly important for digestion. Salivary amylase or ptyalin basically works on polysaccharides. Polysaccharides are complex carbohydrates like starch and glycogen. And in buccal cavity, this enzyme help in 30% digestion of polysaccharides, particularly starch. If I say by the action of salivary amylase, you can see the equation starch is broken down to maltose, isomaltose and limit dextrins. These all are disaccharides. It means maltose, isomaltose and limit dextrins all are made up of two sugar molecules which are joined by glycosidic linkage. But only 30 to 40% digestion occur in buccal cavity. Reason being, food stay here for the shorter duration. So saliva action, it will occur in buccal cavity due to action of amylase, digestion of carbohydrates. That is starch and glycogen begins in our buccal cavity. And after that, we'll swallow the food particle. The food particles not only get softened but also get stick to each other to form a soft ball in our buccal cavity. And that soft ball of food particles, we are calling it bolus. We swallowed bolus, and this bolus then passed through our esophagus, and through peristaltic movements, it is going to enter our stomach. Stomach is a highly muscular bag present in our alimentary canal. It's a J-shaped organ. You can see the anatomy. A stomach consists of four regions basically: fundus, the upper dome-shaped part, cardiac part which receive esophagus. There is main part called body and pyloric part which is connected to duodenum or small intestine. Stomach has strong muscles in its wall. Let me tell you, the muscularis layer of stomach consists of three layers of smooth muscles. There is longitudinal layer of muscles, circular layer as well as oblique layer of muscles. And these muscles enable the stomach to perform churning movements. And these churning movements are helpful for further breakdown or grinding of the food particles. Wall of stomach, mucosa of stomach also has folds and we call them rugae. Gastric rugae are there to increase the surface area to accommodate more and more food within stomach. So stomach has fundus, cardiac part, body and pyloric part. The moment food enters stomach, there occur release of gastric juice, and that gastric juice is released by gastric glands. There are three types of gastric glands mainly found in the wall of stomach, in the mucosa of stomach. These are parietal or oxyntic cells, mucous neck cells, and third one, chief or peptic cells. Parietal or oxyntic cells, they are there to release hydrochloric acid and intrinsic factor. Chief or peptic cells secrete enzymes basically. They secrete pepsinogen and gastric lipase, while mucous neck cells are involved in the production of large amount of mucus. Stomach wall also contains G cells. These are endocrine cells that release gastrin hormones. So gastric glands, which are parietal or oxyntic cell, chief or peptic cell, mucus cell and G cells, they together makes up a juice, and that juice is called gastric juice. Gastric juice is released in stomach. If I say its pH is highly acidic, about 1.2 to 1.8, and regarding its composition, it has water, mucus, hydrochloric acid and inactive enzymes pepsinogen and prorenin. There is one more enzyme present in very small amount in gastric juice. It is gastric lipase. Let me tell you, pepsinogen and prorenin both are inactive enzymes. We also call them zymogens. And since they are inactive, they need to be activated, and that activation is done by hydrochloric acid. So hydrochloric acid, it is activator of zymogens, inactive enzymes in stomach. Not only this, HCl also provide acidic medium in stomach, and that acidic medium is required for the working of pepsin and renin in stomach. HCl is also antibacterial, so it kills the bacteria which are going to enter stomach along with the food particles. So HCl is very important. It is antibacterial, it is activator of pepsinogen and prorenin, and also provide acidic medium for the action of main proteolytic enzyme pepsin. Renin is the enzyme which is found only in infant stomach. It is released by the gastric glands of infants only. As we grow, secretion of renin reduces and altogether stop in adults. If I say function of mucus, mucus is there somewhat providing protection. It protects the lining of stomach against action of HCl and enzymes over there. And the third enzyme, gastric lipase, it is present in very small amount and it is highly sensitive to HCl, and that is why it has negligible action. So gastric juice mainly contain three enzymes. If I say the main one is pepsin, renin found only in infants, and lipase is present only in small amount. This is action of gastric juice. As I told you, pepsin is the main proteolytic enzyme. It is activated by HCl. HCl converts pepsinogen into pepsin, and once pepsin get activated, it is there to break down proteins. Proteins are broken down into proteoses and peptones by the action of pepsin. Renin, I told you, it is the enzyme present in gastric juice of infants. Its main function is to curdle the milk. It is there for curdling of milk. Renin works over milk protein casein. It digests casein and break down casein into paracasein. And that paracasein form calcium paracaseinate in the presence of calcium ion. This calcium paracaseinate is curd. Once this curd is formed, this curd is broken down by the action of pepsin into peptones. So let me tell you, all proteins are finally broken down by pepsin into proteoses and peptones. Proteoses and peptones are partially hydrolyzed proteins. Third enzyme is gastric lipase. As the name indicates, it can work over lipids, but it has negligible action because I told you it is highly sensitive to HCl. So in stomach, mainly food is digested by the action of pepsin. There is no digestion of carbohydrate, no digestion of lipid or nucleic acids occur over here because stomach release gastric juice which has mainly proteolytic enzyme that work in acidic medium. So in stomach, when food get digested, it gets converted to chyme. Chyme is semi-digested acidic food formed in stomach by the action.